Over the past few decades, molecular biologists have developed a number of techniques that can be used to build the structure of the target DNA seed sequence more quickly so as to simplify and standardize the cloning process. Are you familiar with these gene cloning techniques? Let's take a look at the characteristics of these technologies. Restriction enzyme digestion and ligation method. For a long time, restriction enzyme digestion and ligation method are considered to be a traditional cloning method. This method is cheap and flexible, and can be divided into two steps: digestion and connection. Restriction endonucleases can specifically bind to a DNA fragment known as a restriction enzyme recognition sequence and cleave double-stranded DNA. DNA ligase catalyzes the formation of phosphodiester bonds between adjacent 5-phosphate and 3-hydroxylated double-stranded DNA fragments. In molecular cloning, the most useful DNA ligase is DNA ligase from T4 phage, T4 DNA ligase. Gateway cloning technology. Gateway cloning was invented in the late 90s. It was proposed by Wilhout, and was a generic cloning method. Compared with the traditional cloning method, gateway technology has the advantages that you do not need to consider whether the target DNA has appropriate digestion sites, and do not have to have cumbersome and time-consuming digestion and link reactions. Then you can clone the target genes to a variety of target carriers compatible with the gateway technology quickly and accurately according to the direction and reading frame. Gateway cloning technology. It utilizes site-specific recombination integration and resection reactions between lambda phage and E. coli chromosomes, LR and BP. The BP reaction is the recombination reaction to use an ATP DNA fragment or expression cloning to react with a TTP donor vector to create an entry clone. The LR response is the recombination reaction between an ADAR entry cloning and an ADAR target vector. The LR reaction is used to transfer the desired sequence to one or more target vectors in a parallel reaction. The recombination sites ATP, ATTP, ATL and ADAR are specifically recognized by the enzyme inhibitors encoded by phage lambda and E. coli. Gateway cloning technology. Invitrogen has developed expression vectors for different hosts, including prokaryotic, yeast, insect and mammals, greatly improving the applicability of gateway technology. Gateway technology has been widely used in the construction of high-throughput carriers. Gibson assembly. The traditional restriction endonuclease cloning technique will form a scar or seam at the binding position of the two fragments, which may have a subtle effect on the behavior of the DNA fragment. This is particularly a synthetic biologist's headache, because they often want to transform the DNA fragments like promoters and terminators into predictable independent components. Now I tell you about Gibson assembly, a seamless cloning method. Gibson assembly. Gibson assembled was first proposed by Dr. Daniel Gibson and his colleague J. Craig Venter in 2009. Gibson assembly is well suited for splicing multiple linear DNA fragments and, of course, for inserting the target DNA into the vector. First, it is necessary to add a homologous fragment at the end of the DNA fragment by PCR. Then incubating the DNA fragment with a master mix containing 3 enzymes for 1 hour. The master mix contains 3 different types of enzymes. 1. An exonuclease that digests DNA from the 5' end and produces a long sticky end, which facilitates pairing with another homologous terminal. 2. A polymerase used to repair gaps. 3. A DNA ligase to achieve traceless splicing and form complete DNA molecules. see Gibson assembly. The most powerful of the system is that these three enzymes can function well at the same temperature, so the whole reaction can be completed in an hour at 50 degrees. After 1 hour, the sample can be used directly for conversion. Of course, Gibson assembly is also flawed. In the process, restriction site has been generated, and thus it is not convenient to transfer the splice fragments to another carrier. It is advisable to design a restricted site in advance. It should be noted that if the one-time assembly contains more than 5 fragments, the success rate will be greatly reduced. Golden Gate assembly method. The Golden Gate assembly method relies primarily on a type II restriction endonuclease, type ICE, such as Psi and ARI. This enzyme can be cleaved at adjacent positions of its DNA recognition. If we artificially recognize adjacent sequences, we can use the same restriction enzyme to produce different cohesive ends, thereby assembling multiple fragments at a time, which overcomes the limitations of types of enzyme when traditional multi-fragment assembly is performed. Golden Gate assembly method. Specific to the principle, the general endonuclease is divided into 3 kinds. We usually use the second type of enzyme, identifying a specific palindromic sequence, and cutting from them. The Golden Gate generally uses the third type of enzyme, BSMB1, BSA1 and BBS1 to identify a specific sequence, and put several arbitrary bases after a specific sequence. As a result of that, you can overcome that the second type of enzymes must need palindrome sequences and can achieve the connection. Golden Gate assembly method. In addition, we'd like to talk about the connection part. Traditional ligase is T4. T4 can connect the end and the sticky end. But Golden Gate uses T7, which is characterized only to achieve the connection of sticky end. Because of this feature, when connecting, fragments and carriers can be put into a special buffer, where the endonuclease and T7 can maintain a relatively high activity, through the regulation of temperature, to achieve cloning construction. Because the use of T7, so we do not worry about the self-linkage of PCR fragment to reduce the efficiency. Also because it is the use of 3 enzymes, with the design of the sticky end, we do not worry about the random connection of these fragments to achieve the purpose of molecular cloning. Topo clone. Topo cloning uses DNA topoisomerase I, which also has the property of restriction endonuclease and ligases. This enzyme cleaves and reconnects DNA during replication, so that the entire cloning process can be done at room temperature for only 5 minutes. Topo clones do not need to use primers that contain restriction enzyme sequences and can be cloned directly without the need for a linker on the PCR product. Topo clone. Topo Ta cloning principle is same with that of Ta cloning. The only difference is that Ta cloning uses T4 ligase to connect the PCR fragment to the T vector, while Topo Ta cloning uses DNA topoisomerase I. Topo clone. The PCR topo vector is also a T vector, but only a topoisomerase I is covalently bound to its 3 end, and when the PCR product of the protruding at the 3 end is complementary to the T vector, topoisomerase I connects the gap. Since Watson and Crick discovered DNA double helix in 1953, today's DNA cloning technology has developed to such an extent. Synthetic biology is in a period of rapid development, and the progress of these advances will promote the arrival biological revolution. Are you mastered? Source: https://medium.com/ad contact_30600